module here in the Faculty of Biological Sciences. We do cancer biology in 25 lectures and a 2-hour seminar. So you're going to get a much condensed version of that material, but hopefully it still makes sense. But to kind of calm all our nerves, I started out with a nice picture of one of my favorite views in Yorkshire, which is that of Brimham Rocks here. Okay, so let's get started with cancer biology. So here's an overview of the first talk. So I've called this Cancer Basics. It's just some definitions and terms to make sure that we've heard them all. I'm going to talk a bit about the relationship between genes and cancer. Then I'll be talking more at the level of cells and tissue, talking about how normal processes such as tissue regeneration and cell proliferation are hijacked in cancer. I'll then talk a little bit about two really prominent genes, RAS and MYC, and then finish with talking a bit about invasion and metastasis. So first of all, I'm going to start with a definition of what is cancer from a point of view of a cell molecular biologist. So cancer refers to a large number of diseases characterized by the development of abnormal cells that divide uncontrollably, have the ability to infiltrate and destroy normal body tissue. So I'm showing that here in this schematic. So normal cells are organized very well in the epithelium, and they're held together through gap junctions and the other functions that we just heard about. They're arranged into these structures and separated from other parts of the body through extracellular matrix basement membranes. Cancers don't look like this. They are disorganized. The individual cells look different from normal cells, and they're disorganized. They, even at an early stage, can supply themselves with nutrients and get rid of waste by forming new blood vessels. So I'm just going to define a few other important terms. One is neoplasia. It's a term you may hear, and it means new growth. So any abnormal, uncoordinated, and excessive growth of cells is called a neoplasm or tumor. Sorry, just getting rid of something on my screen. It forms through formation of a neoplasm or tumor. So neoplasms, tumors can be benign or malignant. So the difference is that a benign tumor does not have the ability to metastasize and spread or massively invade local tissue. So it's simply an overgrowth of that tissue. And what we're worried about from a disease perspective is more the malignant neoplasms or cancers. And all of these neoplasms result from multiple genetic changes in cells that are irreversible. And I'll tell you more about the mechanisms by which all these happen throughout the two talks. And so cancers are essentially malignant neoplasms, so overgrowth of cells that are malignant, that are disruptive to normal cellular function. So I'll go into this in a little bit more detail. Essentially, everybody has benign neoplasms. Here's an example of just a skin lesion that is quite innocuous. And benign neoplasms, they're non-invasive, so they're localized to a particular part of your body, to a particular tissue. They're well circumscribed, so they're not invading other tissues. They will grow, perhaps, but they will grow very slowly. And they're constrained by local factors. So that's other features of the biology that will prevent them from growing any further. The individual cells and the way they're arranged has some resemblance to the parental tissue. And so an example here is just a mole or a nevus on the skin. Now, a malignant example in skin is quite different. So these are invasive. So there is local spread into surrounding tissue. You can see here. There's also distant spread. Tends to have more rapid growth. And they probably look at a cellular and in terms of the arrangement of tissue, they look less like the rest of the tissue in which they're embedded. An example of this would be melanoma. So you can sort of summarize the differences that a pathologist would think of between categorizing something as being benign or malignant. And some of those I've already summarized, but we can go through them as well. So you can see these very different properties between a benign and a malignant neoplasm that allow you them to be categorized as such by a pathologist. So now I'm just going to talk a little bit about naming of tumors, because I'll be referring to that throughout. And so tumors are named by their classification. So they usually end with the suffix "oma," which tells you this is a neoplasia. And then there's a prefix that denotes the cell of origin. So hepatoma would be a liver neoplasm. Malignant epithelial tumors are called carcinomas. And so these are derived from the cells that cover the internal external surfaces, such as the skin, lung, stomach, and GI, gastrointestinal lining, or the pancreas. And these are the most common types of cancer. So lung, bowel, breast, prostate, most commonly occurring types of cancer. There are also liquid cancers. So those can be leukemias or lymphomas. So lymphomas, cancers of the lymphatic system, and leukemias are blood cancers. These are rarer than the carcinomas. And then, at least in adults, there's the very rare types of cancers that derive from the mesenchymal cells. And mesenchymal cells form supportive tissues such as bone, muscle, fats, blood vessels, and connective tissue. And these are very rare in adults. Now, interestingly, in children, these two rarer types of cancer are by far the most common in children. So adult and children's cancers are quite different in both type and also in the genetics. So leukemia and brain cancers being most common in children. So with that kind of some basic nomenclature out of the way, we can kind of talk a bit more about cancer and how it arises. So I'll start by, again, just reminding you about genes being the basic unit of heredity that's passed from a parent to a child. And that's one definition, but a molecular biology definition would be any interval, any part along a piece of chromosomal DNA that is transcribed into a functional RNA molecule, or that is made into RNA, which is then translated into a functional protein. And it's a very simple language, 4 letters in DNA, but in the human genome, it's very complex, being composed of 6 billion letters, 6 billion base pairs, and 25,000 genes. So every cell has the same DNA, but this is read differently to make a wide variety of cells. So not all RNA and protein molecules are made in every cell type, and the different kinds of RNA and protein molecules that are made then work together to make the structures and the functions of these different cell types. So these are all in normal, healthy cells will make different kinds of proteins. So a central fact of cancer is that it is a disease of the genome. The cancer cells arise due to modifications in DNA, and this can be through mutations, so changes in the DNA from one letter to another, or larger changes. Could also be through the incorporation of DNA, genetic material, from infectious agents such as viruses. In most cancers, the mutation, the change in DNA that's causing it are occurring in the somatic cells, so the majority of the working cells in your body. These are the cancers that are not inherited, and they're called sporadic cancers. But in some cases, the mutations are present in germ cells, and cancer can then be inherited. And so this is why some people and some families have a higher probability of developing specific kinds of cancers. So such cancers are called inherited or familial cancers. It's about 10% of all cases. So I'm now talking about a specific kind of cancer just to exemplify familial and sporadic cancers. And this is retinoblastoma. It's a rare pediatric cancer. It's about 8,000 cases worldwide per year. And it can be inherited, familial, or it can occur sporadically. And familial cases usually occur in both eyes of the patient, and sporadic cases usually occur in only one eye. And the genetic cause is a gene called retinoblastoma, or RB1, that the mutation breaks this gene. It loses its function. And if you look on the right, so what happens with familial retinoblastoma is that the patients have inherited one mutant allele. So we all have two copies of each gene, one inherited from mom, one inherited from dad. And unfortunately, this patient has inherited one mutant allele, but they're okay because the function of retinoblastoma is maintained by the normal copy that's present on in the other allele. The other copy is okay. But that means that all that has to happen for this cancer to occur is a mutation in that one copy that remains. And when that happens, the patient now has two mutant copies of retinoblastoma, and this leads to disease. And this is, it's likely that if it's more like in most cases, it's likely that if this is going to occur, and this is going to occur in both eyes. Whereas in a sporadic case, so the patient initially has not inherited any mutant alleles, and this occurs at some point during their early development. A mutation occurs, but they're still okay. And then a second mutation has to happen. And this is much less likely for two sequential mutations in the same gene to happen in one person. And this is why it's very rare and also very unlikely to happen in both eyes. So I used the word tumor suppressor on that last slide, and I'm going to introduce these two really important terms for two main types of genes that are associated with cancer. And these are oncogenes and tumor suppressor genes. So oncogenes are, they cause normal cells to become cancer cells. They're pro-cancer. And I'm going to give you some more examples of this later on in the talk. Cancers may come to depend on the activity of these oncogenes for their proliferation and survival. This is called oncogene addiction. There are many different mechanisms for converting the normal versions of those genes, proto-oncogenes that have normal functions in the body, into the oncogenes that cause cancer. So mutations here are gain of function. The oncogene is still functioning, but it's functioning in an unregulated way. With tumor suppressors are genes that prevent normal cells from becoming cancer cells, and here is loss of function, such as in the example of retinoblastoma. So the early events in cancer development involve the activation of oncogenes and the inactivation of tumor suppressor genes. So I've been talking mostly about genes, but actually, as I said, the molecules that carry out cellular functions are RNA to some extent, but actually the proteins. So I'll just quickly show you a few proteins and a few examples, again, to give you a scale. So here's a typical small protein. It's very small, 3 billionths of a meter. And this image here shows individual atoms of this protein are shown as little spheres in different colors. So carbon is green, for example, to give you some scale. Now, proteins come in many different shapes and sizes, and they carry out lots of different functions in the cell. And when a mutation occurs in DNA, it can very often alter the shape and the function of the protein that it encodes. And here's an example. So this gene is called EML4. This is a representation of what it looks like. It's quite a complicated looking protein. It's found in most normal cells. It regulates cellular structures. It regulates the kind of internal structures of cells. Perfectly normal protein. This is another protein called ALK. And this doesn't really occur in the adult body. It's found only, it plays an important role in developing the brain and nervous system. It's a receptor protein. And it's important for the controlled proliferation of cells and the development of the brain and the nervous system. So, however, in about 5% of lung cancer patients, there's a major genetic change. These two, the genes that encode these two proteins are mashed together, fused together to create what's called the EML4 ALK fusion. And it makes this completely different protein that incorporates individual parts of the two separate proteins. And this now has the property that it is found in these cells. So this protein is made because it's the control of the production of the protein is through the EML4 part. As this is a protein made in most cells, it's then switched on and made in these lung cells. But the part of the ALK that's fused to it again controls proliferation, but it's lost all of the controlling other parts of the protein. So this protein now is made in the lung cells, and it drives those cells to divide and duplicate, proliferate without any control. And this is an oncogene, and you can know this from, for example, experiments done in mice. So these were mice taken, and the EML4 gene on its own was expressed in the mice. This did not lead to any tumors. The ALK gene on its own was expressed. This did not lead to tumors because it's all controlled. But when you took the EML4 ALK gene, this tumors were grown in all 8 of the mice in which this was put. So I'll change tack a little bit now to explain, well, if, why is this possible? Why is it that these genes can be oncogenes that control, that lead to uncontrolled proliferation? And they're simply, these genes all have normal functions, and they are important, especially in the self-renewal and the healing of your body. So just some simple body facts. In the last 10 minutes, your body will have made 120 million new gut cells, 400,000 new skin cells, and 3 billion new red blood cells. So this is just what's been going on in all of us in the last 10 minutes. So the process of making new cells is completely natural and is absolutely vital for health. Cell division, proliferation is also obviously important in building a body in the first place. So we all start out life as one cell, and each of us comprises around 30 trillion cells, including 86 billion neurons and a similar number of non-neuronal cells in our brains alone. So these processes that built and repaired our body, they're hijacked in cancers. And indeed, cancer is the trade-off for the ability to undergo tissue regeneration. So tissue regeneration allows organisms, people, to have longer lifespans by being able to replace dead and dying cells with new ones, as we all are doing. Now, this has to be very closely regulated. There has to be something that switches on the production of new cells and something that switches that off. And this balance is really important to maintain our bodies. in the aging process, there tends to be tip the balance towards cell death. So this is why certain bodily functions can atrophy as you age. In cancer, the balance is tipped the other way. So there is an increase in cell regeneration, and it's not controlled, and that's why these lumps appear. This is why these growths appear. And this happens in stages. So if we just have this schematic, here's normal cells arranged, normal epithelial cells arranged very neatly, and it's got the right number. You can, in early stages of cancer or early stages of neoplasia, you can have something called hyperplasia. So you just have too many cells, so the cells are proliferating too much. You can then have cells that take on an abnormal appearance. So in this case, their nuclei are abnormal. And then you can have cells that are abnormal looking and disorganized. So what's controlling this proliferation, and why does that go wrong in cancer? Well, each cell has a cycle that it has to go through in order to divide. So we start up here, cells will take on nutrients and will grow, and then they'll make a decision about whether they are going to duplicate their DNA and then go through and divide into two cells. Now, normally, cells only do this in response to growth signals and mitogen, so signals that tell cells that they should divide. And within cells, there are mechanisms that are telling cells to stop, that are acting as a brake on cells growing and dividing. And so this is a very stringent mechanism to make sure that cells only grow and divide when they need to for the body's health. So in cancer, two things are happening. These oncogene proteins become overly active, and they signal within the cell, telling it to go through the cell cycle. And this is happening without any control from outside of the cell. And the other thing that happens is that the tumor suppressor proteins, such as retinoblastoma, that act as a brake, are switched off. And that means that these cells can now just go through rounds of cell division endlessly without any control. So I've been talking about this sustained proliferative signal, these oncogenes that tell cells that they have to, that they can just grow and divide. And this is one of many alterations to a cell and to a tissue that have to happen for cancer to occur. And these changes and these features have been collated and in this fantastic review. And if you only read one thing coming from this, I would recommend reading this. It's The Hallmarks of Cancer by Hannah and Weinberg, where they talk about the different changes. So this is something that we can maybe talk about if there's time or over questions. But as well as cells having to proliferate, they have to evade the suppression of growth. They have to evade the immune system that's trying to recognize them as being incorrect and killing them. They have to achieve immortality, so the ability to divide indefinitely. They have to take on the ability to provide themselves with nutrients through angiogenesis. They have to resist cell death signals, etc. So in general, oncogenes, oncoproteins that are encoded by those oncogenes, they enable these hallmarks, whereas the tumor suppressor acting to prevent those hallmarks of cancer. So I can take a short pause if anyone's got questions, or if anyone just wants to have a little break.
I think, Richard, everyone seems to be okay. So I wonder, should we plough ahead, and then maybe we can have a slightly longer gap before you do your second stint?
Yeah. We can go ahead and do that. Okay,
Wonderful.
so
Thank you.
That's fantastic. Okay, right. So so now I'm going to zero in on two particular oncogenes because they're the most frequently altered in cancer, and they're really central. So if you learn about any two molecules, these are the two to go for. And this is the RAS and MYC oncogenes. So I'll start by talking about RAS. So mutations in the RAS gene are abundant in many kinds of cancer, but lung cancer is particularly abundant. So if we look in lung cancer patients, the kind of key oncogene is one of several where it's known. So many patients, actually, it's not really known. The main genetic change is not really known, but the biggest slice of the pie where it is known is one of the RAS genes. And then various other proteins, including the EML4-ALK that I've talked to you about. RAS is very frequently mutated in lung cancer and colorectal cancer and others. So here's a breakdown of the kinds of mutations that you find. So interestingly, very commonly, the mutation is a change in a letter in the DNA code of this gene that changes one single amino acid in that protein. And it's, you have to reflect on how remarkable that is, that one small change can profoundly affect the activity of this protein. And most frequently, it's in this G12 or glycine 12 residue. In lung cancer, glycines change to another amino acid, cysteine, whereas in other cancers, G12D and G12V are more common. But it's this amino acid 12 that's really important. So what is this doing? Well, RAS is a protein that is really at the heart of cell proliferation signaling. It binds to a small molecule called GDP or GTP. And when it's bound to GDP, so it's guanine diphosphate, it does not signal, it does not interact with the other key proteins that tell cells to proliferate. When cells receive a mitogenic signal, that another protein comes in and switches out the GDP and replaces it with GTP. So this is the same molecule, but with an extra organic phosphate molecule attached to it. And that change switches on the RAS molecule. It now interacts with other proteins that tell the cell to proliferate. Now, RAS has an inbuilt off switch in that it's, with the help of other proteins, slowly hydrolyzes, so breaks off that final phosphate, returning the RAS to the GDP, the diphosphate state, and off. So in a normal healthy cell or tissue with cells that are proliferating, this is what would happen. After the RAS has done its job in sending a signal to proliferate, it will be switched off. This G12 mutations, they break that mechanism. The RAS is much less efficient now at being able to hydrolyze that GTP. So it remains in the on state, and it therefore drives the cells to proliferate, resulting in cancer. And so people like to be dramatic, but RAS is sometimes referred to as being the cancer death star because its activity is so common and so profound in driving cancer. A second oncogene, MYC, is a completely different kind of protein, and it's mutated in a completely different kind of way, but it's just as important. So MYC is also involved in proliferative signaling, but it acts right at the level of binding to the DNA itself. So MYC interacts with DNA and it switches on the expression, the production, the transcription of other genes into RNA and then to make protein. And those are genes that are really important for cell growth and proliferation. And in fact, MYC is in most situations downstream, so the signal passes through RAS to MYC. So MYC is not generally mutated in the same way as RAS, not mutated with a single change. It's very often fused to other proteins. So this means that the MYC, which is normally the production of the MYC protein, is strictly controlled. Because it's fused to something else, it's no longer strictly controlled. And that happens in antibody producing cells, B cells, very frequently. And these cancers tend to be really aggressive. There are 3 MYC proteins, and one is called NMYC, and the amplification of gene of NMYC. So this is where there's now not the normal 2 copies, but there are many, many copies of the NMYC gene present in the cells due to errors in duplication of that gene, replication of that gene. And if that's the case, it occurs in neuroblastoma, so for most common childhood solid cancer, and where that's present, it confers a really poor prognosis for those children, a much more aggressive kind of cancer. So MYC, as I said, acts by binding to DNA and switching on the production of other genes. And for this reason, and again, because cancer biologists like to be dramatic, MYC is often referred to as being the cancer super controller, because it controls the production of many other proteins. So how do we know that RAS and MYC are oncogenes? Lots of evidence, but kind of one standard way is through the expression of the mutated version of RAS, or overexpression of MYC in cells and tissues in mice. It generates tumors. So this is just a schematic of that kind of experiment, but over time, mice that are expressing the MYC gene will, over time, generate tumors. Mice that have a G12 to V mutation in the RAS generate tumors perhaps more quickly, but mice that have both of these mutations will generate tumors extremely quickly. And another piece of evidence is that, so the first RAS and MYC genes were not actually found in humans or even in any animals. They were found in the viruses that cause cancers, in animals, actually in chicken, for example, in the RAS. Many oncogenes were discovered this way. So the virus has a set of genes that's required to copy itself and to make more virus, but it also has somehow hijacked one of these genes from one of its hosts at some point in the past. And now when the virus goes into the host cell, it expresses this viral form of these proteins, and that transforms them. So that's how many of these were discovered. So RAS and MYC are very common, and they're also important because they both act on many hallmarks of cancer. So they don't just affect proliferation, but they also affect apoptosis, immortality, and the ability to evade the immune system. So the fact that there are specific genes that are mutated and on which cancer cells depend gives us a way in to try and develop therapies. When a cell has an oncogene, or when a cancer cell is driven by a particular oncogene, it's usually then absolutely dependent on the activity of that oncogene. The signals, the ability to proliferate and have all the other hallmarks of cancer are the function of that mutated gene. So chemical compounds that can target this oncoprotein have been developed, or different oncoproteins have been developed, and these are some really effective cancer therapeutics in sort of the last 20 years or so, including a BCR-ABL fusion, this drug called imatinib, used in chronic myeloid leukemia, EML4 alpha, I showed you earlier, inhibitors such as crizotinib are used very effectively in non-small cell lung cancer. There's a picture of crizotinib, and with the latest compounds, actually, these patients have a survival of over 6 years, whereas these are very advanced lung cancer patients, stage 4 lung cancer patients will have survival of over 6 years, whereas before these new therapies were developed, those patients would have perhaps 6 months. So this has really transformed cancer therapy. So the same approach has been taken through against oncogenes. Now, until recently, there was no therapeutic to target RAS, but that's very recently changed. And so the mutation that's present in lung cancer, the G12C, can be targeted with sotorasib or other similar drugs. And these are now being used in patients. So this is a huge breakthrough in now being able to treat patients who have RAS mutations. Unfortunately, many of the other RAS mutations, the ones I mentioned, were in colorectal cancer, and also the supercontroller MYC are unsolved problems in drug discovery. So I'm going to finish this lecture by talking a bit about the next steps. So we now we've made cells, cancer cells have been transformed, they are expressing oncogenes, their tumor suppressors have been knocked out, they're dividing uncontrollably. But how do they become then, how do they start to attack the rest of the body or invade? So invasion is the next step in the development of cancer. It's the ability of cancer cells to break out of their niche and invade the local tissue microenvironment. And this depends on several features. So the cells have to become motile, they actually have to move. And this means that you have to lose, normally cells don't do that. So they also have to overcome other barriers, such as the kind of glue that will hold cells together, and the fact that cells don't like to grow over each other in these uncontrolled ways, so there's contact inhibition. Cancer cells will secrete enzymes that will degrade the extracellular matrix so that there's actually space for the cells to go into. And so lots of different enzymes are used in this process. So for this to happen, these are further genetic changes that have to happen in a cell. So beyond what we've talked about already, other genetic changes have to happen in order for this to occur. And then after invasion, there's metastasis, which is how tumor cells can seed to distant organs to form secondary malignant growths. And this happens when tumor cells invade through a blood vessel or the lymphatic lining to be able to then access the rest of the body. And metastasis usually happens in common sites such as the liver, brain, lymph nodes, and lungs. For example, the lung cancer, the ALK lung cancer, very often goes into the brain. And it's really these steps, the invasion of metastasis, that are responsible for the lethal consequences of malignant tumors. And once a cancer spread, of course, it requires systemic treatment. You can't just use surgery to remove them. So let's talk a little bit more in detail about this process of invasion. So for these cells which are in the epithelium to kind of invade, they actually take on the characteristics of the cells that are present in these other parts. So the cells that form these epithelia, these layers of closely packed, organized, and connected cells, these cells have an orientation. They have specialized surfaces on the sides and on the front and on the top. And the internal cellular structures are polarized. So again, they're organized very neatly. So contrast that with blood cells. They're not organized, they're floating around kind of independently. And in general, cells that form the mesenchyme are not connected together or organized. And that's really what has to happen to these cells whose origin is in epithelium is they have to become like mesenchyme cells. And so this process is called epithelial mesenchymal transition. Again, requires further genetic changes for a cell to do that. It's a really complex thing for a cell to completely change its behavior, changes its structure, its behavior. It's actually another normal process during development, but again, it's hijacked by cancers to invade. And in terms of the kind of key genes, these are again proteins that are a bit like MYC in that they change the expression profile so that an epithelial cell that would make one set of proteins that are needed for its epithelial function, instead it changes and it's now making a different set of proteins that make it become a mesenchyme cell. And so here's the whole kind of cascade. You start with a primary tumor where the cells are disorganized. They will then invade into extracellular tissue. They'll undergo intravasation, so they'll be able to enter a bloodstream, travel there to a distant organ such as the brain. They then will move from the blood cell back into the extracellular space, extracellular matrix, and then seed and form a new colony, a metastasis in that distant cell. And that, again, in this part of the process, that requires a reversal of some of those of the EMT. So they're now behaving as more ordered cells that will kind of clump together and form a tumor. So that's where we are. I'm going to stop there for the first half. So do let me know if you have any questions.
Brilliant. Thank you very much, Richard. That was amazing. I'm learning a lot this afternoon, which is fantastic. Does anyone have a question for Richard? Looks like everyone's okay. I mean, so I guess the next stage I was about to say is that I guess you're going to talk to us about how to integrate all of this information into our understanding of tumor growth. But actually, I think Richard has got his hand up.
Okay, thanks, Rebecca. Thanks, Richard. Great presentation. I'm not quite sure whether I missed this but when you talk about people's genetic predisposition to cancer, you talked about the various genes. Do some people have a more kind of general predisposition for cancer? Something the wider immune response, or or their kind of issues with their endocrinology or things like that?
Well, that's a great question. Which I don't really know the answer to. I think I don't know. It's quite a difficult thing to unpick. I mean, the reason that you can get it when it's the level of individual genes is that you can obviously, you can track them. You can see the kind of the frequency and also see how that's inherited and see that it's genetically inherited. So I'll pass on that one.
Okay. A second question is things like smoking, and lung cancer related to smoking, and I suppose other diseases related to smoking. Do certain people have a predisposition to that then that has been identified? The kind of more environmental causes of cancer.
I'm going to talk a bit about environmental causes of cancer in the second part. As far as have people, I'm not sure about that either.
Sorry for asking such difficult questions.
No. You're definitely. These are good questions. I think you need an epidemiologist to answer these questions.
Okay, I'll get back to Amy. Amy gave a great talk kind of last week, so I'll ask them. I'll redirect them to Amy Downing. Thank you.
Are there any other questions from anyone, difficult or or not?
I don't mind easy questions. They're okay.
You're happy with easy ones, are you? Okay, well, they're a bit boring, aren't they? So
no, but difficult questions. I mean, it's good to be, to think about all of these things.
You kind of mind blow. I mean, it's interesting because as kind of engineers and modelers, which is my own background, we do a lot of work to understand kind of spatial heterogeneity within the tumour microenvironment and things like blood flow and delivery of different factors and all this kind of stuff. And it's fascinating to try and think about the link kind of to all of that heterogeneity and the kind of cues that you're stimulating cells with in general, right? And how that links to all these potential triggers that you could then set off that could, and things like, I guess, how tumour cells will ultimately adapt and become resistant to a particular therapy, or after therapy you'll end up with a tumour that's completely, pretty different to the tumour that you started with. So how you kind of
exactly. I mean, so part one, I've definitely presented it like, oh, this is quite simple. There are these few genes that are really,
I know, it's like
and then part two will hopefully blow a lot of that away. But it is, it's really quite, it's an amazing, it is a mind-blowing concept. The idea that, a single change in an amino acid can have such a profound effect in driving cancer. And yet the cancer itself as a system is so unbelievably complex, and it has the evolution. And when you look at all the other mutations as well, it's there's probably a word for it, things that are simple yet extremely complex at the same time.
Is there I presume there is, but I obviously I'm not an expert in these areas, but is there a link between other kind of inflammatory diseases and cancers? I'm sure there is, but are they well characterized, or I presume that probably depends on the specific disease and all sorts.
Area of research, the link between inflammation and cancer, it's very, very clear. So so it's a whole thing that I'm not really talking about, but in the second part I'll talk a bit about mutation. But actually, a lot of the mutations that cause cancer are present in normal tissue, and as the ability to kind of do the sequencing and find this out, it's sort of shocking how prevalent these mutations are. So that alone is not enough, and it's thought that the kind of the presence of inflammation is then a driver for those mutations to be able to kind of come through and express themselves and really for the cancer to really take off. So it's a really active area, and I think that would be it'd be a great thing to kind of include in the discussions. Is this, I absolutely would not want to give a talk on inflammation and the complexities of that, but there's lots of good people around who could do that.
That might be a really interesting topic for a seminar, I guess. Maybe we can report that back to the report. Maybe we'll link that back in with the OncoEng teams for a future seminar. But okay, brilliant, thank you. Oh, there's a question from Ashleen on the chat. So, beyond the hallmarks of cancer review, do you have any other recommended reading? I mean, I've read the hallmarks of cancer one. It's excellent, isn't it?
Lots.
Where to start?
Trying to think where to start. That is a good place to start. I think it would depend on what aspect of this you want to get more into.
Would you be happy for anyone to drop you an email, Richard, if they've got kind of specific questions or advice? Okay.
Yeah, definitely.
So Ashley, maybe that might be if you were happy to drop Richard an email for. There you go. Great, thank you, Richard. She says, brilliant. Okay, fantastic. So we've got Richard up for our next talk. He's obviously going to be carrying on to tell us the link between everything he's just told us and tumour growth,